Hello everyone, so I'm just going to do a video on the Punnett squares and how to use them. If you're not already familiar with the basics of Punnett squares and genes, you're definitely going to want to brush up on that before watching this. Basically, you need to know that about recessive and dominant genes. So if you have a specific genotype with a dominant gene, so let's go with hair for our example here. Brown hair is dominant, so we'll put that as a B, but it's a capital B, and it's a capital to show that it's dominant, right? And say we have a pair of those genes, so this is the genotype. If we have a pair of those, then that means that it's got both dominant brown hair genes in there. So that means that the person who has this will have brown hair. Say for example, if we have red hair, someone with red hair has to have two of the recessive genes, right? Because what we find is that if they have a dominant and a recessive gene in that pair, the dominant one is called dominant because it takes over. It's the one that is expressed physically. So you can have the gene for both of them, but if one of them is recessive and the other one is dominant, only the dominant one will be displayed. So there we go. That's something to keep in mind. Which means that if they do have the physical characteristic of the recessive gene, that means that they can't have the dominant gene in there. Otherwise, they'd be displaying the physical characteristic of the dominant gene instead. So this, if someone has red hair, they have to have double recessive. All right? So if you're not familiar with that, and that really confused the hell out of you, so maybe brush up on the concept of dominant and recessive quickly before watching this, but that's basically what you need to know. So let's go through this. I'm going to go through the possibilities of this example here. So if we have these genes here, what are all the different combinations that you can have? So we've got double B, we've got BR, and we've got RR. And let's do a few examples of the combinations you can make. Let's maybe change that to blue. Alright, so let's say our parent one is homozygous dominant. So homozygous, by the way, again, just a refresher, homozygous means like homo isn't the same. It's only got one type of gene in there, homozygous. So this is homozygous for brown hair, homozygous brown hair. And this one down here is also homozygous because it's only got one type of gene in there. So it's got homozygous red hair, right? That's the genotype. We've got two of the same gene, it's homozygous. And zygous comes from zygote. So when the gametes join together, they form a zygote, and that's talking about the way that the chromosomes form, join together from the haploid cells. But anyway, without digging too deep into that, let's talk about these still. So homozygous, if it has both of the same. So this one, heterozygous there. Hetero, meaning like one and another type of one. So two different, it's a mix of different things. Hetero, right? Homo, hetero, and homo again. All right, so let's go with, let's say, hetero there. And our thing that we do is just take the top one and the side one and put it into that square. So this one, the top one is up here, and this one, the side one is down here. So let's go that way. Up here, B, and over here R. Over here B, and over here R. All right, so these are the possibilities of the children. So we have B, B, and B, R. What we notice there is that with this combination, there's no possible way to get this. There's no possible way to get that homozygous red hair, right? And because that red hair is recessive, and we have it in this heterozygous pair with a dominant gene, then that recessive gene is not going to be expressed, right? So what we see is if this homozygous dominant, homozygous dominant is 50% of the children, and this heterozygous dominant is 50%, but they're both brown hair and neither of them are red hair, what we get is that while this is the genotype, 50% homozygous blue, homozygous brown and 50% heterozygous brown, the phenotype, I really can't write very well today, can I? The phenotype as opposed to the genotype, the phenotype is the physical characteristic that is expressed. Phenotype. And what would the phenotype be? The physical characteristic that's expressed doesn't take into account the mix of genes, it just takes into account how they're expressed. Let's just nail that point home. How they're expressed, brown hair, brown hair. Phenotype is 100% brown hair. Cool. So that's that example. And let's sort of get rid of that one there. Let's show a different example. And then we might move on to the questions. All right. So let's say, for example, go with this here. So we're taking that one and that one for this one, this one and this one for that one, this one, this one, this one, and this one. So you get the point there. If we take that possibility space for the children, now we're seeing BR and RR. And now BR, there's 2 of them out of 4, which means there's a 50% chance. And for that, it's a 50% chance of homozygous red. So we've got heterozygous brown and homozygous red are our 2 possibilities. So what's our genotype for that? So we have dominant and recessive there, which means that the dominant will overpower. So it's 50% brown hair for that one. So brown, 50%. And now what's this one here? That's homozygous recessive, the red hair, which means that we're going to get red hair expressed. So that means that we have brown hair is 50%, and red hair is also 50%. So really hammer home the fact that that phenotype is just the physical characteristic that's expressed. So we're looking at what color is the hair or the eyes, or how tall is the person, right? So we're looking at what is actually expressed, and the genotype is what genes are involved in determining that. Right, so it's a different thing. Phenotype, genotype, they're related, of course, but they're different. So let's sort of move on from there. Let's go to an actual example. Maybe we can just get rid of that. There we go, that's better. All right. So in watermelons, solid green rind color, G, is dominant to stripes, which is lowercase g. So let's go with solid green is capital G, and that's green. Well, actually, let's just write it as solid, just so we remember solid versus stripes. So solid. And then we have lowercase g equals stripes. So again, our possible genotypes here, that's heterozygous. Heterozygous for solid, because what's going to be expressed, the phenotype is that it's going to be solid, because that G for solid is dominant over that recessive. So that'll be solid. Then we have homozygous solid. And we have homozygous stripes. Alright, so now we can look at the question. A farmer crosses 2 watermelon plants that are heterozygous for rind color. Heterozygous for rind color. So what's our heterozygous one? It's the dominant and the recessive together. So for heterozygous, it has to be 2 different ones. Why did I put a B for that one? God knows. Who knows? Alright, so G, 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 G. All right. So we can see here from our thing here, farmer crosses 2 watermelon plants that are heterozygous for rind color. both of them are heterozygous, what are the odds that the offspring will have solid green rinds? So we have solid green there because it's two things. So let's write our probability space out here. So we've got, that is 25%, right? And we've got that one and that one, the heterozygous ones. There's 2 out of 4, which makes it 50%. And then here we've got homozygous recessive is 25%. Now this one here is going to end up with stripes. So stripes, what's that, 25%? Are there any other that make stripes? No, there's not. So that's 25%. And then the other two, this one, homozygous dominant, of course, is going to end up with the dominant one, which is solid, which is 25%. And then this one, the heterozygous, the genotype is also going to be solid. Now, there's no difference in the phenotype between these two because phenotype is just talking about what's expressed physically. So the thing that's expressed physically is solid rind color, which means that our phenotype for solid, the probability is just 75%. Because 3 out of 4 of those give us a solid rind color. Cool, so that's our answer there, 75%. Solid green rinds. All right, so let's try another one here. This is our last one for this video. So in mussels, brown coloring is dominant, right? So let's go with B, brown. Right, and blue coloring is recessive. B, blue. Easy. So lowercase for recessive, uppercase for dominant. A homozygous brown mussel crosses with a blue mussel. So homozygous brown, homo, they're both the same, homozygous brown, crosses with a blue mussel. So a blue one. If its phenotype is blue, and blue is the recessive, then that means that we can't have that dominant gene in there. We can't have that in there because if it wasn't there, it would be brown instead of blue. We know that if it is blue, which is the recessive, it has to have this genotype, right? Has to be both recessive. So we know that when it says blue mussel, it's both recessive. So with that out of the way, let's try this. So cross that and that, cross that and that. Same deal there and there. And there and there. Right, so what percentage of the offspring are expected to be blue? Well, let's get rid of this for the time being, and let's write over here like we have been doing. So how many of those are the heterozygous brown? 1, 2, 3, 4. Out of 4, 100% are going to be heterozygous for brown, which means that the phenotype is also going to be brown 100%. So where does that leave us for blue? How many of them are blue? What's the percentage that are blue? Well, 0%. Because this doesn't give us anything that results in our double recessive. And we need that double recessive to be that physical characteristic having been expressed in an organism. So, for example, if we didn't have the double recessive, we gave it a dominant in there instead, then the dominant one would take over and it would be brown instead of blue. So there's no way to have that and still be blue. Blue has to be that. So yeah, that's all the examples for now. That's a basic introduction to the Punnett square concept. This is one I'm going to be running through in another video here. And it's a bit complicated just because it takes a bunch of different applications of that same thing in the same problem. So it can be a bit difficult since you're putting layer on layer on layer. So I'll go through that in a separate video just to give you a break. But yeah, that's how you do Punnett squares. Basic Punnett squares.